Hello everyone, thank you all very much for coming. I'm Lizzie, and as has been mentioned, we have come all the way from the real Cambridge over in England. We're pretty hard to miss in our lime green t-shirts. We're very excited to be here today. We've been working all summer on a project whereby our aim has been to design new tools for self-organised pattern formation in free-swimming bacteria. Now, we've designed two possible systems which could be used to test these tools. The first of which is a system which is comprised of bi-directional signalling, and the second system is a population-based bistable switch, which will be discussed in the future work section of our presentation. So the bi-directional signalling system is illustrated in this cartoon on the overhead. It comprises two cell types, type 1 and type 2, which are active simultaneously. The type 1 cell produces 3-oxo-C6 homoserine lactone, which the type 2 cell responds to by the expression of GFP. The type 2 cell, in contrast, produces 3-oxo-C12 homoserine lactone, which the type 1 cell responds to by expressing red fluorescent protein. So that's just a very quick overview of our system, which we're going to go into in more detail. This is the format of our presentation. We're going to discuss the genetic basis of the tools we designed. Then we're going to discuss the modelling of our system. And finally, we'll describe to you the experiments which we've done during the summer to test the tools that we've made. So now I'm going to hand over to Nicholas, who's going to describe to you the tools which we've designed.
Thank you, Lizzie. I will now describe the proposed system for bidirectional communication in greater detail. Sorry. Yeah, this diagram essentially captures the essence of what's going on at the molecular level. There are essentially two plasmid-bound genotypes capable of communicating with each other. Both are analogous in that each of them contains a sending circuit and a receiving circuit. The novelty in our approach lies in the fact that for the first time we propose to use two independent quorum sensing systems in parallel. We have the Lux system from Vibrio fischeri and the last system from Pseudomonas aeruginosa. So let's start by looking at the sending circuit in more detail. Now, what does the sending circuit contain? The sending circuit contains a promoter which has leaky expression and resulting in a basal level of transcription of Lux-I protein. This results in a low intensity signal source of 3-oxo-C6 homoserine lactone. This molecule then goes and binds to the Lux-R protein, and this forms a transcription factor complex which goes and activates transcription from P-Lux, resulting in autoinduction. And finally, positive feedback leads to the promoter being locked in a state of maximal activity. We have a similarly analogous system in the type 2 cell that you will recall from the previous diagram, which also contains a sending circuit and a receiving circuit. The type 2 system is distinguished by the fact that it has the last system, and therefore it produces 3-oxo-C12 homoserine lactone. This is also a diffusible signal molecule, which goes and binds to the last R receiver in the receiving circuit of the former cell. This response can be directly visualized by fluorescence, and this response is furthermore customizable. Now, the fascinating property of this system is that the diffusibility of the signal compound or signal molecule allows the study of population-wide signal integration events. The sending circuit is the most important part of our system. And we wanted to engineer a variety of properties in this sending circuit. For instance, we wanted to be able to control the otherwise spontaneous autoinduction. Furthermore, we also wanted to be able to tune receiver sensitivity. Therefore, it was not possible to design the sending circuit using biobrick construction, and we had to synthesize this sending circuit de novo. I will now explain what are the properties of this sending circuit that are so unique. Consider this slide. So this is an example of the Lux autoinducer. The last autoinducer is analogous. The Lux autoinducer contains the Lux-I gene and the Lux-R gene in a reverse orientation behind a double promoter. Generating this double promoter and this reverse orientation using biobricks construction would not have been trivial. Now, furthermore, we introduced a left promoter, which is P-LAC, which is repressible and by LAC-I molecule, which can be encoded for by a gene on another plasmid, allowing further capacity for signal integration. Furthermore, this LAC-I repression is relievable by IPTG, as you all know. We also introduced 4 unique restriction sites that can allow this promoter, or each promoter, to be replaced, that can allow the introduction of another gene behind LUX-R before the 5' terminator. Therefore, from this very simple device, you can construct a whole variety of other devices. One important thing about this construct is also the codon-optimized nature of the coding sequences of LUX-R and LUX-I, which is far more important in the case of the last autoinducer to allow stable expression in E. coli. The last autoinducer is analogous, except the identity of the repressible promoter differs in that it is PC1 lambda. So to summarize, what are the essential properties, or what are the key features of our system? Consider this. First and foremost, what we propose to do is to study population-wide signal integration events as opposed to the direct interaction of molecules in a single cell. Therefore, we are observing non-cell autonomous behavior. Also, for the first time, we propose to uniquely link two independent quorum sensing systems, that from Vibrio fischeri and that from Pseudomonas aeruginosa, allowing the cross-wiring of the LAS and LUX system. Therefore, each cell type or each cell type, plasmid-bound genotype is capable of both sending and receiving at the same time. You can consider this to be a sort of having like 2 phone lines, one for making calls and one for receiving calls. This allows bi-directionality of communication. Furthermore, these systems, as you would notice, are analogous, and therefore essentially contesting populations are equivalent. Also, as I mentioned, we've introduced repressible promoters, and this repression can furthermore be relieved. Therefore, we can tune signal kinetics, and we have tunable signaling kinetics. Also, you would recall that the receiver response, in this case we are visualizing as fluorescence, is customizable, so we can replace it. And therefore, there is potential for feedback coupling of the receiver response to the sender circuit. And finally, I would like to reiterate that this system allows complex signaling dynamics to be directly and vividly visualized.
Thank you very much, Nicholas. We're now going to hand over to Charles, who's going to discuss the single and multicellular models which we have designed for us.
Thanks very much, Lizzie. Thanks very much, Nicholas. Hi, everyone. I'm going to talk about the models that we built for our system. So the first thing we did is trying to establish a single cell model for our system. A single cell model essentially describes what's happening inside the cell, i.e. the transcription or translation activities. So here is our type 1 cell, which our model is based on. What we're particularly interested in is to observe behaviors such as autoinduction and switching within our system. So a few assumptions we made is that we assume both types of cells have identical dynamics in that they are equally efficient in transcription and translation, and they respond equally well to homoserine lactones. And the second major assumption is that these genes, LUX-I and LUX-R, are constitutively expressed. So after we derive a set of equations, we put them in the MATLAB, and here's a plot that we generated. So on the vertical axis is the concentration of molecules, and on the horizontal axis is the time domain. As you can see, there are 4 curves here, and the 2 blue lines here, they represent the sender circuit of our system, whereas the 2 red lines here characterize the receiver circuit. So if we just take a look at the blue lines for now, this dashed blue line represents the level of homoserine lactone, which is made by type 1 cell. And we can see it adopts this sort of sigmoidal type of curve where it has this exponential increase initially and tailing off at the end. Same as this one, which is the amount of LUX-I protein that is made within the cell. So these 2 curves essentially characterize the autoinduction behavior of the cell. So now if we forget about the blue lines and look at the red lines, what happens here is that we intentionally put a step input of the opposing homoserine lactone into the type 1 cell, and what we observe here is a switching effect, which is shown here. So here's the concentration of RFP in the cell, and we can see it's increasing rapidly over this range. So the conclusions that we draw for our single cell model is that the system is workable and it shows stable behavior, and provided we have a threshold of opposing homoserine lactone, coming into the cell, we should be able to switch the cell by turning its color. So after we completed the first model, we actually took a step further and tried to model the system as a whole. So we want to see what's going to happen, how the system is going to look like on a plate. And it all started with the idea of Conway's game of life. So Conway's game of life, as I'm sure some of you have already heard of it, is basically a game for bacteria where the cells are set to live in these sort of rectangular grids. And we set up a couple of rules to them on how they can replicate and move around and diet on these grids. And their behavior, the cells' behaviors are actually largely dependent on their neighbors' behaviors. And this is where we think this idea is appropriate basis for our model, because our system is essentially about cell-to-cell interaction and communication, and cells' movement largely depends on neighbors as well. So there's a strong analogy between Conway's game of life and our system. So if you imagine the Petri dish is composing of thousands of these grids, we can pretty much get a simulation of the cell movement. And again, we made a few assumptions here. So here's a simple illustration of how the simulation program works. So initially, we have these 4 inoculations here. The two cyan ones represent type 1 cells, and the 2 blue ones represent type 2 cells. And over time, these cells grow, replicate, and move around on the plate. And bear in mind that at the same time, these cells are synthesizing homoserine lactones, and these molecules diffuse out, whereas these cells themselves, they're trying to swim towards each other. And this is sort of pattern that we would expect to get eventually. What happens here is that at the intervals where these 2 types of cells meet each other, there's a change of color. So the blue ones turn into green, and the cyan ones turn into red, giving us this cross pattern. And lastly, I would like to show you a video clip. So the whole thing was implemented in C++, and basically what happened here is there's an inoculation of type 1 cell in the middle, which is cyan. It's not very clear. And surrounded is type 2 cells, which is blue. So over time, these cells grow, and as we can see, there is a change of color at the boundaries where the 2 types of cells meet each other. So the blue cells turn into green, and the cyan cells turn into red, giving us this sort of flower pattern. So I guess the real system would differ from this simulation program in that the whole process will take much, much longer, and also that we do not necessarily have to expect the change of color at the boundaries where the cells meet. So it could have happened well before that. Thanks very much.
Okay, so thank you to Charles. We're now going to head on to our final section, which will be presented in English, to discuss the experiments we conducted over the summer and the future work which we're hoping to do.
Now, to verify whether our system would indeed work in living cells, we had to conduct a series of experiments. Because we proposed to engineer our system in free-swimming bacterial cells, the first logical thing to do was to standardize the swimming assay. So we tried a variety of conditions, and finally we determined that E. coli swims in an optimum nature in 0.3% Pacto agar on a plate being incubated at 30 degrees Celsius overnight. Furthermore, we tried a variety of E. coli strains and determined which one is the best swimmer. So if you consider this diagram or this picture, we have E. coli MC1000, which covers a much greater distance in the same amount of time than MG1655 and XL1 blue, even though it is swimming in less than optimum conditions. Furthermore, we found that if you have multiple populations or multiple inoculations of bacteria on a swimming plate, this leads to very interesting patterns. For instance, consider this, where you have 4 inoculations of XL1 blue cells on a swimming plate. Notice also this very interesting clear zone of separation that results between 2 opposing populations. To examine this, we studied this under a wide field inverted microscope, and we indeed found that there doesn't seem to be any bacteria in this region, possibly due to nutrient depletion. Furthermore, we found that if you have differently motile E. coli cells on a swimming plate, then the potential for pattern formation is even more interesting. And therefore, we can conclude that differential cell motility leads to spontaneous pattern formation. We then realized that these patterns can be visualized in a vivid manner if you transform the E. coli cells with fluorescent biobricks. And here you have red fluorescent MC1000 and green fluorescent MC1000. I suppose you can all admire the beauty of these patterns. Then, The next thing we wanted to do was, because our system relies on lactone signaling, we wanted to test these lactone producers and receivers in the BioBricks registry. So what we did was we transformed E. coli cells with these lactone producers, and we used a novel assay described by McLean et al. To test whether these E. coli cells were indeed producing lactone. So here you have this agar plate, which contains a top load of CVO26, Chromobacterium violaceum. And you inoculate this plate with a strain of E. coli MC1000, which is producing homoserine lactone. CVO26 responds with violacin production, which can be easily and vividly visualized. This here is a control inoculation of a strain not producing lactone. Furthermore, a modification of this assay means that it can be used to distinguish between AHL molecules with N-acyl side chains from C4 to C8 in length and C10 to C14 in length. This assay can also be used to have some fun, as this picture would illustrate. Unfortunately, it's not very clear, but you can get the gist of it. We then found that if the interaction of AHL sender cells and receiver cells on a swimming plate with cell motility defining the zones of response can lead to very interesting patterning, as this picture would illustrate. So what you have here is a sender cell in the middle and receiver cells surrounding it. All these cells consist of the same genotype, but only those which are in the proximate zone can express the homoserine lactone molecule. And this, can express GFP in response to the homoserine lactone molecule. And this is due to cell motility. Also now, I would like to discuss future work, because we believe that this, that coupling these two quorum sensing systems, from the last system and the Lux system, can be used to generate a population-based bistable switch. So if you have a plate and you inoculate it with two populations of isogenic bacteria biased into two different gene expression states, and these gene expression states can be visualized via the expression of green fluorescence and red fluorescence, then over a period of time, these cells will replicate. And a chance event would result in green cells probably outnumbering the red. This means a few red cells will convert. Feedback would result in more conversions. And finally, green cells would dominate the plate. As you can see, this has great potential for artificial pattern formation. We have modeled this system, and we believe that it can indeed work. And this is the proposed genetic circuitry for this system. I would be happy to explain this genetic circuitry if you have any questions regarding it. But now I would like to discuss the applications of the myriad systems that we have discussed, or the tools that we have made for engineering self-organized pattern formation. Pattern formation is a critical aspect of development, and therefore these tools can be used to understand development. Tissue invasion and metastases both depend on position-dependent gene expression and differential cell motility. Furthermore, biofilm formation, especially in Pseudomonas aeruginosa, which is where the last system originally comes from, depends on lactone signaling. And of course, artificial pattern formation has great potential for use in tissue engineering. With that, I'd like to hand over to Lissy.
Yes, so we'd just like to finally thank very much to our supervisors, Jim Hasselhoff and Jim Escherichia, for all their help and support this summer, and also to our generous sponsors who have made our project possible. And finally, we'd just like to have a mention for our friend Kai, who did the project with us, but unfortunately couldn't make it today due to passport problems, so couldn't take part in the presentation. So thank you very much.
Looks like we have 6 minutes for, 6 minutes for questions, and I want to ask the first one, which is, which was your favorite part that you selected for the competition, and why did you select it?
So we selected the auto-inducing devices, or the auto-inducing cassettes that I described, the Lux and last cassette. And the simple reason we selected it was because of all the different properties that it allows us to generate, as I described in the presentation.
Other questions?
Oh, and we also selected 2 other parts which we made for, which can possibly be used to extend free antibiotic assembly. So, put that in there.
Okay. Questions? Back?
Yeah, what's crosstalk like between the two quorum sensing systems?
Yes, that is a very interesting question, and the point is that it is critical to our system that they are independent, and they do indeed seem to be quite independent in nature. The Lux and the last system. In fact, we tested the last system from the BioBricks registry, and we found that it doesn't seem to really work, which is why codon optimizing the coding sequences of the last systems as we did in our constructs is quite important for stable expression.
So the characterization of the auto-induction system, was it mainly done in swimming assays, or
yes.
did you also do it in homogeneous environment where you could control?
No, we primarily use swimming assays, and our proposed system is to be engineered in free swimming bacterial cells. But there is potential for trying other methods.
How did the team collaborate on this? Were there different tasks that were divided up, or did everybody work on the same thing? Did everybody have the same expertise?
Basically, we have 3 engineers and 3 biologists on the team, so it's pretty good balance. But everyone involved in the lab work, and part of the team were working on the model. So yeah.
And back, so your multi-population model, does it involve just the spread, the division and spread of bacteria that you observe different, the appearance of different colors, or does it also
Could you speak up a little, please?
in your whole cell cell population model,
Yeah.
when you observe different colors, is it due to the movement of bacteria, or also to the movement of thought and?
That's a very good point. Well, the switch of color is due to both the diffusion of observed electrons and also the swimming itself of the cells. So as the cells are swimming towards each other, homogeneous electrons are synthesized as well, and they try to diffuse too.
So you model these 2 diffusions simultaneously?
Yeah.
Question here?
In making the bi-directional operator, where you could insert control in the feedback, you mentioned in passing during the construction of that you ran into some limitations with the BioBricks assembly process. I've never heard anybody criticize that before. And so I'm wondering, in developing a solution for that where you sort of reorient one of the existing BioBricks and putting it into, did you think about developing a sort of scalable approach that would make the BioBricks assembly system more generically useful, or did you develop a sort of ad hoc construct that solved the problem for your one particular construct?
So for this construct, we essentially got it synthesized de novo.
Yeah.
So we didn't, because we thought that reversing the BioBricks and orienting them in this fashion to generate a double promoter would be quite complex to do.
A second, just minor follow-up question. When you were doing the swimming assays, I noticed you were going across different concentrations of agar.
Yes.
Did you ever look at different sources of agar? It's been reported, for example, that if you buy the agar from iGEM in Japan versus Vibrio and whatnot, you get very different effects.
That's right. So what we did, but the iCAN agar effect has mainly been reported for the swarming assays as opposed to swimming. And we did initially try to get some swarming assays to work as well, and it didn't work at all in bio agar for E. coli. And we tried to do it for using iCAN agar for E. coli, but unfortunately we were not able to. I mean, we did see some distinct effects, and we did see some kind of swarming in iCAN agar, but it was not as vivid or as interesting as the swimming assay. So we thought maybe we can try it later, or let it be for the moment.
One last question, 1 minute.
I was wondering for this flipping, can't you use the not one sites in the BioBricks?
So you're right, there are those knock-on sites in BioBricks, but in the flipping, what I basically was talking about is that we've introduced 4 unique restriction sites here, which are different from the BioBrick restriction sites. So you've got BAMIT, I'm sorry, I've noted down the exact restriction sites, which are, so basically in order there are SPH1, BAMIT1, BSP1, and SAL1, which are all different from the BioBricks BioBrick restriction sites. So therefore you can actually use this as a BioBrick construct, because it fits the requirement.
Those not one sites, you can invert the
Yeah, but the problem was that you inverting this promoter using biobricks would have been quite difficult.
Okay, let's thank the Cambridge team again.